There are two concurrent workshops. One is the cloud computing, the other is single cell data analysis. If you are interested in the cloud computing, please use the Zoom link in the chat. If you are interested in single cell data analysis, just stay here. Thank you. And good afternoon, everyone. Welcome to a single cell data analysis workshop. I'm Mingao. I'm from Informatics Institute at UAB. I'm the host of this workshop. The instructors for this workshop include the Dr. Chen, me, and Dr. Thanh, Nguyen, Dr. Zongliang Yue. And here shows the schedule of the workshop. And the first speaker is the Dr. Chen. He is the Associate Director of Informatics Institute and Professor of Genetics and Computer Science. He will give an overview of the computational advance and application trend of single-cell genomics. Then I will introduce how to work on the single-cell RNAseq dataset, including do the demo for using the Python package Scampi to work on the single-cell RNAseq dataset. Next, Dr. Thanh Nguyen will focus on the mining spatial pattern in single-cell data analysis. And the last one is the last speaker is Dr. Zongliang Yue. He will show how to use the Seurat work on the single-cell RNAseq in the Seurat interactive app. And we will have 10 minutes for the question and answer part. If you have questions, you can put it in the chat. And you can find all our slides in the UAB box. Now let's give time to Dr. Chen.
All right. Let me share my screen. I hope you can see my slides, right?
Yeah.
Yeah, so I'm not sure if I can finish in 20 minutes, but I'll try. I've got more slides than is needed. So what I'll do is to give an overview of single-cell transcriptomics analysis overview. And here's my outline. I'll start with cellomics and single-cell analysis basic overview of concept. And then I'll go to a little bit of a concept in data preprocessing, why we need to do it. And then I'll go into dimensionality reduction and clustering. And the last, I will talk about applications. So it's not meant to be comprehensive because nobody can do a lot in 20 minutes. It's meant to get your feet wet and get you interested in what the single cell analysis is all about. So we are familiar with the genomics. There are 21,000 human protein coding genes and 3.2 billion base pairs. When we do the tissue transcriptomics, we usually think about just 4 major tissue types and then the 80 human organs. We treat them as if they're homogeneous, but we all know in biology that's not the case. But when you try to go down the hierarchy and try to explore the detailed human cell subtypes, you realize that there are more than 200 cell subtypes. So if you actually go chase every individual cells in a human adult, and that number become mind-boggling. If you want to multiply that with the number of genes or even base pairs, and this quickly becomes just mind-boggling. But this is at least what the single cell analysis is trying to do, is to get some heterogeneity survey of the tissues at the level that is more fine-grained than the tissue themselves. So looking at the bulk RNA-seq analysis, the workflow is very straightforward. You get the tissue, you extract the bulk RNA, do the average gene expression from all the cells, and you don't actually get to see the heterogeneity unless you actually get sub portion of the tissue. But if you do single cell analysis, you can separate the cell into individual cells and put them into sequencing. You can actually see in each of the cell type or individual cells, there are different proportion of gene expressed, and therefore you can understand the heterogeneity. That's really the one benefit of doing single cell analysis. But of course, the ultimate science that we're trying to answer is a lot more sophisticated. And I would term this word cellome. Essentially in single cell analysis, we want to answer these biological questions, such as how are cell types arranged, how are cells represented by its individual omics, and the transcriptomics is one of the major part, but it's not all. And how does a genomic variation in the cell lead to variation changes to the cell function? I think this morning you already hear from one of the keynote talk on the clonal evolution of cancer cells. And then how is the tissue developed, and then how do cells communicate with each other? So collectively, this becomes more complex, but have direct application in precision medicine and drug discovery. Okay. At the national level, there's huge project going on. And I think it's called the Human Cell ATTIS project. And the idea is that you go to different tissues and then you do these single cell analysis project. And they're different cells from different organs, but then they may share cell type. And then you build a database that pull all of the cell types and gene expression signatures into a huge database. So in the future, if there are new data coming in of unknown tissue of origin, you can project it and then you can sort them into hierarchies. And then you can unambiguously say, well, what kind of tissue this is and where does it sit in the context of existing knowledge of human cell ATTIS. But for individual investigator project, we're not lucky to have that kind of a funding. Usually we start with a sample of some kind of a tissues. But the process overall roughly can be simplified as this. You isolate the cells into droplets, wells, tubes, or whatever you have. You lyse it, do reverse transcriptase of mRNA into DNA, cDNA, and then you barcode them, amplify them, or in some cases you can pull them before. And then you do deep sequencing, and then you just try to reconstruct the cell origins, figure out what is the proportion of the genes in there. But there are lots of the informatics problem right there, right? It's different batches, you may create variations, so-called batch effects. When you don't capture all of the cDNAs, or then you have a capture efficiency problem. When you amplify them, certain strand may be amplified differently. You have amplification biases. And when you do the sequencing, depending on the sequencing depth, you also suffer from potential false negative situations. So, so I want you to keep that in mind. And here's an overview of single cell RNA-seq analysis pipeline, which I believe that the rest of the tutorial will give you a very good idea. And you start with the data acquisition, and then you do data cleaning. I'm going to talk a lot about data cleaning. And then there's a main analysis that involve clustering, classification, trajectory assignment. But today I'll just focus on the clustering. And then there's a lot of main analysis that's more downstream. I believe Zongliang, our fourth speaker, will mention a little bit of that in his tutorial. Okay, here's an example what you can get in the end. If you do a very comprehensive 98 cell type single cell analysis, put them together, you can actually do this clustering. Each color patch represents the cell type. So it's very nice that based on all the gene expressions, then you can still, even with the drop off of genes, and you can still unambiguously assign them to the right cell type with the right knowledge. Now let's talk about the denoising and imputation in the pre-processing step. So RNA-seq, single-cell RNA-seq data is a lot noisier than bulk RNA-seq data. So here's one figure. If you look at the single cell, which is a purple bar chart, when you put them into different expression group, the curve bend more to the rightwards, which is actually more proportion of zeros than the bulk RNA-seq. So there's a more zero, meaning that you measure them, somehow they don't show up. There are many reasons that there's zero value gene expression. Sometimes they're true zeros, but sometimes they're just dropout events. And also you see the pink ones are the single cell analysis, and the green ones are the bulk analysis. And the x-axis is the variance. The variance tend to be a lot higher in the single cell analysis result. It's probably a good thing because you're capturing the true biology of heterogeneity. And I listed all of these potential reasons that can generate stochastic process of a gene expression that make every single cell to be somewhat different. Most of it is a biological, but in some cases, it's a technical noises. Okay. And then there's also a trade-off between read depth and the cell coverage. We usually have limited budget, and when we do sequencing, and there's just a certain trade-off what we have to do. Let's say we have all of these, and if you do fewer cells, and maybe each cell you can do a higher read coverage. If you do more cells, and then a lower coverage per cell, that's gonna actually affect the quality. Okay. In the single cell RNA-seq analysis, pre-processing mostly is about removing poor quality cells. For example, you can do histogram like this to plot the count of cells that has how many genes covered. And suppose your cutoff is here, and everything that has less than 2,000 genes per cell, for example, you just cut it off. Or you can just use a cutoff based on the reads. Map the reads. If it's too low, then you can just trim the cells. But the trimming cells dropping cells is not the only thing you do. You also, for comparison, you have to normalize them. And in many cases, you also have to only look for genes or features that are significant. But then there's another issue of impute dropout values. Suppose that you have a lot of zero, and the zero is in the right gene that you think you know for sure it's a CD4, it's in the CD4 positive cells. It cannot be dropout. How can you just fill out the dropout value? That's what the imputation is all about. In this software called the iImpute, they are actually looking at all of the similarity of other cells based on the query cells. And then the query cells for each gene, they're using a previous program called the Saver that predict using the pink value, the pink color, what is the likelihood that this dropout is due to, this zero is due to dropout. If it's dropout, then it's not a true zero, then you can predict it. But the prediction is look at this cell and look at all the similar, just correlated the cells, and then you average all the value and get the new value assigned. So the algorithm is seems to be pretty straightforward, but it's a very useful tool. Now, as with all the bioinformatics software tools, we should not assume that any software would produce the same result. This is a paper published recently in Briefing in Bioinformatics. They look at more than 19 tools. And then 17 were, 19 tools. And you can see some of these tools seem to just separate these synthetic data set and clusters very well. Some of them are really mixed. So choosing your tool carefully is very, very important and critical even. And then I won't go through the details, but they even go through like a very careful grid to say in what scenario, what quality control issues, and what software seem to do better. So Saver, the one that I mentioned earlier, which is based on models and seem to do very well. And that's why I was citing that iImpute, which is based on derivative of the Saver software. Okay, so I think I'm gonna quickly talk about the dimensionality reduction and clustering. One of the major tasks in the single cell analysis is to convert each cell, which is represented by several thousand genes in a high dimensional space, into a lower dimensional space so human can visualize and explore it. So this is a paper published that visualize all of these different cell types. You can see that you can group them based on different mesenchymal cells, immune cell, and then other cell types, and then at different stages even. It's a B cell, pre-B cell, pro-B cell, neutrophils. And this all relies on a software called embedding, so multidimensional embedding software. And talking about embedding, one of the easier ones that everyone understands is a principal component analysis. It's a linear method. It just look for the axis of the variance and then to give you the new coordinate system and then give you the so-called principal component. But the problem of this approach is that when the data is highly dimensional and then somehow folded in a space that is highly interdependent, then you're getting this highly weird, skewed structure that you cannot separate the cluster well. So you can't really use that linear method. So people come up with a nonlinear method. And the basic concept is you have this multi-dimensional called a data manifold that looks like a Swiss roll. And instead of measuring A to B's Euclidean distance, you actually follow the trajectory of the data. And then as if you're flattening out the surface of the data fold and measuring this distance at a lower dimensional space. That's called a geodesic distance. And TISNI, one of the major software, uses stochastic neighborhood embedding. Essentially, first to look at multidimensional conditional probabilities between the two, and also come up with a lower dimensional counterpart and try to define this distance between them and use the optimization techniques to minimize the distance of matches between the multi-dimensional projection to the lower dimensional. And the T part really comes when you use a traditional normal distribution, the data could still be congested. But then if you use a T distribution, and you can actually flatten the curve a little bit to avoid the congestion of the data. So it spaces out nicely on the space. And so this is one example of a TISNI result. And you can see each dot is a cell, and you can color them with gene expression of this particular gene. In general, t-SNE is not very stable, but if you actually selectively only tease out some of the genes, like in this embedding, you don't have CD33. In this embedding, you tease out 4 more. And it seems that the embedding is more or less preserved from one to another. So that was a nice feature at the very beginning of the analysis. But t-SNE is not the holy grail. One of the problems with t-SNE is it doesn't have the domain knowledge embedded. So there's a software called a Similar that essentially use a gene expression matrix, gene expression to cell matrix, goes through multiple kernels to define to cell similarity matrix. And each of the kernel can be, okay, my favorite pathway as a bridge to define this cell-to-cell similarity, or maybe using only disease-specific, cell type-specific genes. But you can build these kernels and use weight to build this similarity matrix, and then you do embedding, and the result is going to be improved from the pure, straightforward t-SNE. This is something that the people usually don't do unless you work with like a great informatician. I think my time is okay. No time left? Really? Okay. Okay. Then I think I'll just quickly finish up. And then there's the global structure with techniques that's called a UMAP. And UMAP can not only look at the local structure like t-SNE, but sometimes you have very similar cell type. They're not, well, actually dissimilar cell types, and then they're all congested. So you may want to use a UMAP instead of a t-SNE to do it. And there are even better algorithm that's called a TriMap that give you better result than either t-SNE or UMAP. And this TriMap is based on more advanced mathematics, and I don't have time to go through it. Am I really out of time, Min?
Yes.
Okay, then I won't go through the application, but I'll leave the slides just in case that people are interested in afterwards. Thank you.
Thank you, Jake. Great overview. Because we need to balance the audience, there are only 10 persons in the cloud computing workshop. Could someone move to the cloud computing workshop? Thank you. Jake, can you stop sharing? Yeah. Next, I will show how to work on the single cell RNA set. Now I will share the slides. Can you see the slides?
Yes.
Thank you. Hello, everyone. This is Minggao. Welcome to the single cell RNAseq data analysis workshop. I'm from Informatics Institute. Today, I will talk about the single cell RNAseq data analysis. This is the outline. At first, I will give a short introduction and overview, and then I will show the different steps for work on the single cell RNAseq dataset. And finally, I will do the demo for analysis of single cell RNAseq data, use the Python package Scampi. As we know, the fundamental unit of an organism is a single cell. Each cell or cell type may have distinct function. The research on single cell provides great insight into our understanding of biological diversity and the mechanism of some disease. The traditional experiment only work on the bulk samples. We can only know the average gene expression of the samples, which may lead to ambitious cell-fit decision. but use the single cell technology, we can know the expression of each cell and know the different gene expressions of each cell and know the different subsets, which can help us understand the population heterogeneity. Single cell affect diverse areas, including the neurobiology, cancer biology, immunology, microbiology, and some other areas. Single cell technology are popularly used in cancer genomics. That we can use the single cell technology to generate the different data sets, such as the genomics data, transcriptome data, or proteomic data, epigenomics data. Use this data set, we can analyze by the different by informatic method. We can identify the clusters, lineage, or network, which in turn to generate the new biological hypothesis. This biological discovery in turn guides the development of the new technology and the new bioinformatic method. Also, the single cell technology popularly used in immunology. For example, the single cells can captured by the different method. After sequence of the single cell, we can detect the gene expression, and also we can know the different subpopulations of the cells, and even we can trace the cell process, use the trajectory analysis. Also, we can use the single cell VDG-seq technology to know the more information about the VDG-seq, about the T cell receptor or the B cell receptor information. Here shows the different method of single cell environment. The key technology that allowed jump in environment scale, or jump to hundreds of cells were enabled by the sample multiplexing, and jump to the 10,000 cells achieved using the integrated fluidic circuits, and a jump to several thousand of cells using the liquid handle robotics, and with further tens of thousands cells enabled by the random capture technology, use the nanodroplet or the pico-well. And the recent study have used the sealed barcoding technology. We can work on the hundreds of thousands of cells. Here shows the cell numbers were reported in the different papers by the date. And the key technology are showing here, just like the SmartSeq2, DropSeq, InDrop, or 10x Genomics, or SeqWell, and other technologies. The most popular used single cell platform is the droplet-based single cell platform. Here shows the basic workflow. We can prepare the cells from solid or liquid tissue. Use the droplet method to split the cells. Then we can prepare the CDAs from the cells and send it to do the sequence. After we get the sequence read, use the bioinformatic method to analyze. We can address lots of biological questions. Here shows the popular platform, 10x Genomics single cell platform. Use this platform, we can do the single cell RNAseq, single cell DNA-seq, single cell VDG-seq, or ATTIS-seq. With the development of single cell technology, we generate many single cell data. That is an opportunity for us to get great biological discovery. At the same time, there are challenges written, such as the basic programming needed to interpret the data set. Also, the information contained in the single cell data needed to transfer into the relevant biological knowledge. That needs the benchwork scientists and bioinformatics scientists work together to figure out. Next, I will introduce the basic step for work on the single-cell RNAseq dataset. The first step is from the raw rate to the gene expression matrix. Here shows the sequence data from the 10x Genomics single-cell platform. We get the raw rate that's shown here that contains the cell barcode and the CDA sequence. We can use the cell ranger to process the data set. The step that's shown here, we can map the FASTQ files to the reference genome and remove the duplicate UMIs and sort the cell barcode, and then count the reads aligned to each gene and to each cell. Then we can get the gene expression matrix. Here shows the gene expression matrix that shows the cells, and this is the genes. The number indicated the expression of each gene in each cell. And here shows the command for the cell range count. Here we can say here's how to use this command. We should label the output name of the samples, the path of the FASTQ files, the prefix of the sample name, and the reference genome directory. Here shows the examples that include the output name of our samples, and this is the raw FASTQ files. And here the prefix of samples, and here shows the reference genome directory. After we run the cell range count, we can get this output. This includes the summarized files. Based on the summary files, we can check the mapping ratios, how many cells were counted, how many genes per cell, and also we can get the aligned BAM files, and we can get the gene expression matrix. The gene expression matrix can found in this directory. We can upload this directory to the other tools such as the Scampi or Seurat, and to do the further analysis. The first step we process the gene expression matrix is for the QC and the filtering. The goal of this step to remove the low quality cells and potential doublet. And there are many parameters we need to consider, such as the UMI distribution, number of genes detected, or the percentage mitochondrial genes. Usually the high number of genes detected in the cells, that means sometimes the doublet. And usually the high mitochondrial gene associated with cell death. So we need to filter these cells. After we do the filter and the QC, we need to do the normalization of our dataset. The goal of this step is to make the profile of each cell comparable. The simple approach is to scaling the library size to some value such as 10,000. Here shows the dataset before normalization and after normalization. Each comment to regress out the effect of number of UMI and percentage of mitochondrial genes. Next, we need to do the dimension reduction. For this step is to visualize the structure of our data. The PC is one of the techniques used for to do the dimension reduction. Dimensions means the numbers of features of our dataset. And why we need to do the dimension reduction, because the larger dataset needs the more computational power and time, and the visualization is impossible for the larger dataset. So we need to do the dimension reduction. After we do the PCA analysis, we can do the clustering for our cells. The clustering is to assign the cells to group of similar cells. Here list many methods that can be used to clustering the cells. The popular one includes the Scampi, Seurat, SC3, and T-scan. In the following slide, I will introduce the Scampi do the clustering. And after we get the clustering, we want to know the marker genes for the cluster. So we need to do the differential equation analysis. This step is to find out what's the difference between the clusters. There are lots of options. There included the pairwise comparison and also the such as class A compared the class B, and also include the marker gene identification. That means such as the class A compared with all other clusters. Sometimes we can get a long list for the candidate marker gene, so we need to use the p-value or log fold change to filter these marker genes. Here list the many methods can be used to do the differential equation analysis or identify the marker genes. The popular ones such as the Wilkinson, that is the default used in the Seurat, and also popular used in the Scampi for identify the marker genes. And also the single cell RNAseq dataset can be used to do the trajectory analysis. In single cell trajectory analysis, pseudo time is measure how much progress of single cell make during a process, such as the cell differentiation. From this plot, we can see the cells in different time points located at the different positions of the trajectory. We can check the gene expression of the cluster in different trajectories. And for the multimodal data analysis, that include one popular used method, that is single cell VDG-seq. Single cell VDG-seq can be used to identify the VDG gene usage, and you will know the more information about the both chain of the receptor. Both include the T cell receptor or B cell receptor. Here shows are examples. We use the sample from the B cells from B health control and the SLE patients. We did the single cell RNAseq and single cell VDG-seq. We want to compare the usage of IgH gene in health control compared with the SLE patients. We compared the IgH genes in the patient in SLE patients and health control use our in-house method. We can say there are some IgH gene significant different used in the SLE patient compared with health controls. Next, I will show you how to work on the single cell RNAseq dataset using the Scampi. Scampi is a Python-based single cell RNAseq data analysis toolkit. It can be used to for the processing, visualization, clustering, or identify the differential expressed genes or the marker genes, and do the trajectory analysis. Also, Scampi can integrate it with other tools to do the data analysis, such as the velocity analysis and the trajectory analysis. You can get more information from Scampi website. Now let's show how to use the Scampi work on the PBMC cells, single cell data set. We can get the PBMC single cell RNAseq dataset from the 10x Genomics website, or we can use a Linux system. We can download, use the following command. We can create the directory and go to this directory. We can use the wget to download the dataset, and we can uncompress the dataset. Also, you can go to my GitLab, and I have put everything in this link that includes the dataset and the tutorial and the Jupyter Notebook. And next, we need to install Scampi. Here I install the Scampi using the through the Python. First, we can install the mini conda 3 at first. We can use the wget, download the mini conda 3 package, and then we execute the bash files. And we can, when we finish install the mini conda 3, we can create the conda environment for Scampi and install the install conda and related package. Use the following command. You can also get more information in my GitLab link showing here. And now we can create a new Jupyter Notebook using the Scampi conda environment. Then we load the, at first we load the Scampi and other related package just showing here using these functions. And then we can make this system settings, and then we can load the PBMC dataset using the read 10x matrix function in Scampi package. We can, after we finish this step, we can check the dataset showing here that contains 2,700 cells. And the next step, we do the cell filtering and cell QC. At first, we keep all the cells contain 200 genes, and all the genes can expressed in 3 cells. We do the basic filtering, and next we count the percentage of mitochondrial genes. We can use this plot to show the total count per cell, percentage of mitochondrial gene, and number of gene detected. And we can find the outlier for the percentage of mitochondrial gene is about 5%, that's shown here. And find the outlier for the total number of gene in each cell, about 2,500. So we can use this outline to filter our cells, just showing here. The number of genes detected, we use less than 2,500, and the percentage of mitochondrial gene less than 5. Next step, we do the data normalization. We use this function in the Scampi package. We can just show you here to do the normalization. We use the library size is 10,000 shown here. And after normalization, we need to detect the high variable genes. This can use for our clustering step. So use this function, we can detect the high variable gene is shown here, and we can plot the high variable gene shown here, and the black dot indicated the high variable gene, and the green dots indicated the housekeeping genes. Next, we can do the scale the data and remove the uninteresting source of the variation. The source we wanted to remove include the total count per cells and percentage of mitochondrial gene. We use this function in Scampi to do this step. And then we can use this function, scale function, to scale the data set. After the scale the data, we can do run the PC, use this function in the Scampi package, and also we can visualize the pieces of the dataset. Use this plot. This can let us know how many pieces we should consider in order to compute the neighborhood relationship of the cells. Here shows the 10. We can use the 10, the number 10. PCs for the neighborhood graph preparation. And so next we do the cell clustering. We compute the neighborhood graph of the cells, use this function. The number of neighborhood we use 10 just from the PCA plot. The value is from the PCA plot. And then we can embed the neighborhood graph in 2-dimensional UMAP using the UMAP and use this function shown here. And next we can do the clustering. For the clustering step, it's popular use the London method in this package. So we can do the London clustering, use the function just shown here. We can see we get the 8 clusters from the data set. And now we can visualize the clustering results using the plot UMAP function in the Scampi package. And we can show the UMAP plot that contains the different colors indicated the different clusters. There are total 8 clusters for the PBMC dataset. And we can also check the expression of the targeted gene in the cluster using the function Scampi plot UMAP, such as the color indicated the expression of the targeted gene, such as the CST3. There is high expression in these clusters, but lower expression in other clusters. Next, we can detect the marker genes for each cluster. At first, we can rank the marker genes using the different method. That here shows we use the t-test method to identify the marker genes. And then we can use this function to plot the top marker genes. Here shows the top marker genes in each cluster. We can also try use the different method to identify the marker gene. Here we use the Wilkinson method to identify the marker gene. Here shows the top, here plots the top marker gene in the 8 clusters. We also can use the different methods to visualize the marker gene. We can show the top 5 ranked markers in each cluster using this function. Also, we can use the Violin plot to show the marker genes. Here, the group indicated the cluster, the N genes means the number of genes we want to show. Here shows the top 8 genes in the cluster 0. And we can also use the Violin plot to visualize the marker gene. Here they are the marker gene. They are the marker genes, and the color indicated the expression of the marker gene in different clusters. Here shows the expression levels of the marker gene in each cluster. And based on the top marker gene information, we can annotate the cell type to each cluster, such as we can use the R7R gene to assign the cluster 0 as the CD4 T cells. Also, we can use the CD20 gene to assign the clusters 2 as the B cells. Based on the marker gene information, we use this function. We can assign the cell type to the cluster. This use this function in the Scampi, and then we can use the UMAP to visualize the annotated clusters. That's showing here. The UMAP indicated the different clusters, the annotated clusters labeled with the different colors. We can use the different method to visualize the marker genes in the annotated cluster. Here we use the dot plot to visualize the markers. This is the annotated cluster. Here shows the marker genes. The colors from the white to the red indicated the expression of the marker genes from lower to higher. And the dot side indicated the percentage of cells in the clusters. We can also use the Violin plot to visualize the marker genes. We can use this function to make the Violin plot, and here also show the marker genes expression in the annotated clusters. Now we can go to the my GitLab use this link. I'll show the more details about this analysis use the Jupyter Notebook. Can you see my GitLab?
Yes.
Thank you. Here include the data set, and this is a quick start. This includes the information about the system requirements that need the Linux or Python and Python. And here shows install the Scampi through the conda. This command shows how to install the conda install the Scampi in the conda environment. And you can get the directory use this command. And also this Jupyter Notebook here shows this Jupyter Notebook shows this tutorial. Here is the Jupyter Notebook showing how to use the Scampi work on the RNAseq dataset. First, we can load the Python Scampi package and other package. Also make this system setting, and then we can load the PBMC dataset. And we can check the dataset showing here. That's how many cells showing here. And next, we can do the filtering step. And the basic filtering, and we count the mitochondrial gene, percentage of mitochondrial gene, and then we plot the mitochondrial gene, the percentage of mitochondrial gene, and the total cell, to the detected of genes in each cell, and we find out the outline, and to filter the cells. And next, we do the normalization, use this function, and then we identify the high variable genes. Use this function, we can identify the high variable gene, and then we can plot the high variable genes. So here, next we regress the uninteresting variation, such as the total count and the percentage of mitochondrial gene. And we can scale the data set, use this function. And then next we can do the PCA analysis, and we plot the PCA and get the numbers we will use for the compute the neighborhood graph. And this step is to compute the neighborhood graph. We use the numbers of neighbors is 10. And then we can embed the graph in the 2-dimension, use the UMAP, and do the clustering, use the London method, and we can identify the 8 clusters. And we can use the UMAP to plot the cluster result, and showing 8 clusters. And also we can use UMAP to identify the target gene or the marker genes expression. Use this function. And next we can identify the marker genes for each cluster. We can test the different methods to identify the marker gene. Here we use the t-test. It's we plot the top marker gene for each cluster shown here. Also, we can use the other method to identify the marker gene, such as the Wilson method. We identify the marker gene and plot the marker gene shown here. And also we can use other method to visualize the marker genes. Just use the list to show the top marker genes in each cluster, and use Y-line plot to plot the marker genes. And based on the marker gene information, we can assign the cell type to the clusters. We use this function to annotate the cluster. And we can plot the annotated cluster shown here. And we can visualize the marker gene, use the either annotated cluster, use the different methods, just as showing in the use the dot plot showing here. And use this dot plot function in Scampi. And we can also use the Y-line plot to visualize our marker gene in the annotated clusters. And also we can save the data set. You can get all the information about the tutorial from this GitLab link. And finally, thanks other instructors in this workshop, and thanks all the members in the ATTIS program committee, and thanks for our collaborators. And if you have questions after this workshop, you can contact us. Or if you need the bioinformatics support, you can contact us. I have left my email and Dr. Chen's email here. Thank you. So next speaker is Dr. Thanh Nguyen. He is the postdoc in Informatics Institute from Dr. Chen's lab. His talk is about the mini spatial patterns in single-cell data analysis. Now let's give time to Thanh.
All right, thank you, Min. And let me post the link for the resources in my talk. Give me a minute. So, okay, link to data. Okay, let me, here we go. Okay. So, hi everyone, I hope we have a good ATTIS day so far. And I'm talking about a relatively new and slightly rare topic in single cell analytics, which is about the spatial pattern. How do we find them? How do we define them? And how do we use them in and get something useful in single cell? So in this talk, my objective is, to give everyone a relatively good understanding about what spatial really is, and how we can get the spatial. They are original definitions or original designs of how to compute the spatials, but that is a relatively expensive, experiment to do so. So we later in our lab in the Informatics Institute, we define our own pseudo spatial and try to use a pseudo spatial to and to solve two problems in single cell, which is one is a cluster marker and second is the gene correlations. using the spatial or pseudo-spatial information. So, number one, what spatial is? It is space. So where does this come from? So there is a Nature paper. I put it over here. When people want to do, they have the tissue image. That is the visual cortex of the mouse image. If the mouse and they capture a snapshot of this tissue, and they do they select a little region over here for the staining image. So from here they got a staining image, and they use a microscope to see a large number, a lot of cells over here, which is like zooming, I would say 100x, and they can see many of the cell type, in just a small piece over here, and they seem to be in different location in this small snapshot. So what does this mean? That means even if you take a very small piece of the tissue, that can be homogeneous, and the homogeneity of the single cell or the cells in the tissue could be could be detected or could be analyzed by the locations of those cells in the tissues. So we can say, well, like over here, this region, you can see the glutamine RN and then GABA being interacted with a few OPC cells over here. This doesn't, this region doesn't have. So we can analyze cell-cell interaction, hopefully, in a small regions, and we can try to find out the signaling gene or signaling pathway of that small region if the staining image is designed to highlight the interested genes we want to see. So the summary, to do that, we need at least a cross single cell RNA-seq, certainly. The second thing is the histology. So you choose people over here, they choose a few genes that they want to observe under the microscope, staining image. That staining image gives us space and resolution, but they don't tell us anything about expressions. So we need single cell there. Sometimes people can add the bulk RNA-seq from the same tissue. What it means is, over here, they can further dissect the tissues into small region and take those regions out and do sequencing in the bulk way. So that will further increase the quality. Why do they do so? We'll see that later. But you can imagine that is a costly operation. So basically, the two main components, we have histology, the staining image, and single cell, and we have to map them together. So we can take the light intensity, which is a basics idea. Because we do the staining image, we have light intensity of the marker gene, which we want to observe under the microscope. And then we also have the gene expression from the single cell. We take the same gene expression, we map that to the same gene marker light intensity. Then we can approximately, identify the location of X cells. In case we want to increase the mapping quality, we can do bulk RNA-seq. So we take those little, the little piece of the tissue. We do the bulk sequencing. We only look at the marker gene from the bulk expression, and that map together with the single cell expression. So basically, the outcome, we can look, those data set and the image. You can go look at the source over here, and some of them are available in Seurat. But after we get all of those things done, like analyzing single cell and analyzing the the staining image, we can see at least three things. Number one is a distributions of, likely the location of certain cell types, the ligand. For example, here we can see the ligand genes as concentration and predict these, cell type producing ligands, and then see where they are in the tissues, and then see, and then that is a ligand. And then we also do the same. We can do the same thing with the receptor over here, and then we can, form hypothesis of, cell-cell communication signaling pathway. Now, in case we don't have the funding to do such an expensive operations, we can, in our institute, we try to make the thing simpler, and we ask ourselves, what, can we really use the, clustering, including the embedding these new maps, the locations of the cells in the embedding space instead of the tissue image. So the prior analysis, like over here, it says yes, that is possible if we take a look at the cluster shapes and cluster density and define our spatial, I would say pseudo spatial that way. So that matters because, like for example, here, this one is our collaborators' data set. The tissue is the peripheral blood. So we have that is the neutrophil. So you see there is a long tail of neutrophil, and that highlight a subset of neutrophil. So shape, irregular shape like long tail matters. Sometimes, in many cases, it highlights a subset of cell. Or you can see over here that cluster, long cluster 7-3, they imply other cell type rather than the monocyte macrophage. Even though in the embedding they look attaching to each other. Or you can look at on the other hand, you look at the B cell number 5, that green cluster over here, and we look at this, the density of the cells. Then we see those, even though it doesn't seem to be a irregular shape as that part, but we can see the higher concentrations over here, higher density of this part. So that also implies something. So to do, to represent the density and shape of the cluster, we define our polygeny framework. So basically, the polygeny is a way to, characterize a cell subset by, in our version 1.0, which is used for marker, cluster marker. So we define the whole cluster over here, including red and then cyan as a background, everything in the cell cluster. So you can, and then after that, if you project every cells in this background on the x-axis, you expect a relatively like half of a circle distributions. And then if here, the cyan is a cells, they are cell expressing a certain genes, and we also project them onto the x-axis. We can expect like somewhat skew distributions because they are more dense over here. So we do so, and we can do that in all of the angles in the 2D space from which the embedding was from. So we can do the same thing all over, for all angles from 0 to 60 degrees, and we can have multiple, for each angle we have one histogram for the foreground here and one histogram for the background. And then for each distribution like this, we can compute the Gini, which is the inequality that distribution represent. And after doing so, we can plot them into a polar curve for each, for each subset, for each subset, such as the whole cluster here in red. We project them into an angle, and then we have one distribution. From the distribution here, we compute an angle, and then obviously for the whole cluster, because of the circle shape, we have a circle, and then for this foreground, the cyan here, the shape is quite different. So we have a shape, in not very smooth like this, like the cyan one, and then the next point, how do we define the markers here in the cluster? So our hypothesis is if a gene is a marker of a cluster, then the two curves, the cyan here, which is the gene expression, which is a cell expressing the gene, and the background, which is a whole cluster, has to be very closely and similar. So this is one example. So we plot, here is the UMAP representation of chiromyocyte. ACTC1 is a chiromyocyte marker. We plot these two, you see they are quite fit together. This one is not a myocyte marker. So we plot those two, and we see the cluster red, and then the cell expressing this gene, they are somewhat far away from each other. And the second problem we can try to solve for is to infer the gene-gene correlations. So basically here we have the neutrophil. We see 3 genes here, and then the expression plot of those 3 genes by themselves already show that we have 2 type correlation here. So NPG and LTF, they express highly in the same kind of cells. So we have strongly correlation, and then NGP and CSF3R, they are like complement to each other from the expression plot, so they are negatively correlated. So we can plot those polar gene. We see, so this curve and this curve are quite similar and overlapping. So we have positive correlation, and then this curve and this curve are, very different. So they are somewhat negatively correlated. And then we also define, to quantify how the two curves are similar, we can compare, we plot them together, and then we can take the bounding curve and the intercepting curve for these 2 genes, and get the over, so we have here is like the covering curve for these 2, and then the intersecting for these 2. So intersect divided by the cover small, and we have a low correlation. Over here we have the strong correlation. And so we have the manuscript being accepted in Genomics, Proteomics, and Bioinformatics for the first idea, polar gene curve, and I upload it's over here. We have a website. So this explain how to run our a tool in MATLAB. Yeah, there's a tutorial here. But in case if a little difficult to do so, and you don't have MATLAB, then we also provide window application and Linux application of those. They have the installer inside, so you can download them, run them away. I would recommend a good GPU computers and everything being proper to make the app run smoothly. But basically, you can see here we have the expression. It's like a matrix. We have the coordinate x and y for all cell. We have the cluster ID for X cells, and we have the gene list. The gene we want to investigate. We would load them into the app. So I would load that one is the coordinate. The 2D coordinate, and then the expression, and then this list of gene symbol, which is here, and then the cluster ID, which is here. So after loading everything, we can click on this to plot the cluster. This, for my machine I'm using, it is a little slow because of improper configuration. But you can see things showing up. You can view those. It is like cluster 1, cluster 2, 3, and so on. Here, you can type in the gene you want to show and the cluster you want to analyze. I can change this to MYS7, which is another markers for cardiomyocyte. So, so this MIS7 is highly expressed in this cluster, which is number 1 here. So if I plot, sorry about the machine. That is not a good machine to run this demo because of the improper GPU configurations. And I don't have a better machine else. Oh, here it goes. So you see these two being fit together. If I change the cluster here to 3 and I replot, so you see you see different curves. You might also, if you have a good GPUs and a properly configured machine, you can even save the mouse around this area and click on save file, and you can save the image like here, for example. But I guess I don't have the best machine with myself at this point. So this is the last thing I would show in the tutorial. And last but not least, I would like to acknowledge other people working with me, Dr. Chen and Zongliang. They are all here, and the collaborator is from UAB Dental School, Dr. Ping Zhang. If you have any questions, please feel free to ask. I think by the end of this sections. Thank you very much.
Is there any question? Thank you, Sai. We can move to the next speaker. The last speaker is Dr. Zongliang Yuan. He is a postdoc from Informatics Institute, also from Dr. Chen's lab. His talk about single cell data analysis using the Seurat and interactive Seurat app in the Ubright tool.
Hello, everyone. So good afternoon. I'm Zongliang from the Informatics Institute in University of Alabama at Birmingham, and today I'm going to talk about the single cell analysis using the Seurat and also interactive Seurat app. As you already know that from the previous speaker, the single cell pipeline is usually follow those the first a 6-step procedure, but afterwards we will also introduce some downstream analysis like the pathway analysis and gene network analysis. Afterwards, we are going to introduce about the Azumad interactive app, which is provided by the official Seurat lab, and the Ubright tool, which we have developed, also involve our architecture, develop like a lot of developer team here. So as you may already know that the flowchart of the single cell RNAseq analysis, the upstream is usually a cell ranger procedure that already have been discussed by Dr. Gao, and afterwards we will just use the downstream analysis such as Seurat. The input of this is the matrix MTX, features TSV, and barcodes TSV, and this is the general RNAseq analysis workflow. So once you're working with the Seurat, currently they have updated to the version 4. You can install from CRAN that using this command line, install package Seurat, and after you installed it correctly, you will load this library Seurat successfully. Afterwards, they also provide this Docker that if you want to just migrate the same environment they are working, you can use a Docker pull from the official website here, and they also have the latest link here. The first step is set up the Seurat object. So after you have the upstream analysis result, you need to provide the directory here and use the read 10x if you have the 10x data. So for example, I'm using the example of the peripheral blood mononuclear cells, PBMC, freely available from the 10x Genomics. There are 2,700 single cells that were sequenced on the Illumina-Seq 500. The raw data can also be found here using this link. And afterwards, once you load the data, you need to create a Seurat object using this command line create Seurat object. And here you can also provide a project name, like here, because this is around 3,000 cells, so it's PBMC3K, and the minimum of the cells is 3, and minimum feature is 200. And after you successfully build this object, you can also have a view of what the matrix looks like using this command pbmc.data. And for example, they can provide you these 3 samples, the gene samples, and also the first 30 cells here. So after you just run that command, you will get this matrix, and the dot values means there's 0. Second, you will do the cell quality control. That is the filter cells based on the criteria here. So basically, there are 3 criteria. First is to detect the mitochondrial gene expression using this percentage feature site. And after that, you will send back to the original object PBMC and give them this object called the percent mitochondria. And also you can also view the Weldon plot, the overall distribution of the N features, RNA, N counts RNA, and percent of these mitochondria. So N feature is the unique molecular identifiers in each cell, and N count is counts the total expression value in each cell, and mitochondria is the percentage of the expression of the mitochondrial genes divided by the overall genes expression here. afterwards, you can apply some features according to the plot. Also, you can view this feature scatter plot by do this two-dimensionally. For example, you can just use the N count versus the percent of the mitochondria to see the overall the correlation, and also the N features versus the N count, and always this is a positive correlation here. And after you decide what the parameters you want to use to filter out this noise, for example, here they use the N features RNA larger than 200, N features RNA not too much with the expression, because there could be a diplet scenario here, so it's less than 2,500, and mitochondrial expression is less than 5. The third step, the normalization, you will use the command to normalize data, and here is to make the profiles of each cell comparable, where our previous speaker already introduced. So give you overall the skill. So for the first, the raw data, you will see this is left skewed data distribution for the expression. But after the normalization, you can see every cells have a very comparable for each genes, and also the overall distribution is normalized. And of course, you can do the detection of highly variable genes here. They provide this called find variable features, and you can provide the top N features to be marked as a red plot here. And also you can use the label points to just see the top 10, like for example, they provide these top 10 gene symbols to be added in this scatter plots as well. The scaling of the data here, they have provide the all genes as the feature to just scale the data. Of course, if you want to do some short time running, you can also have a 2000 as the default to run this scale data process. In the step 4, perform linear dimensional reduction PCA. So you will, in the Seurat, you will run this run PCA and also use the variable features here. And after you run the PCA, Seurat always also provides you the top N features to be displayed here. For example, if I use a 5, I will see the 5 top positive genes correlated to the first component. Of course, you can have multiple dimensions. So 1 to 5 is the first 5 dimension to be shown here. Visualize the PCA, you can also have multiple ways. The first is to do this Vis-Dime loading, which you can see that rows represent the genes here, and the x-axis is the positive, whether a positive or negative correlation in this percentile component. And also in the dime plot, you can also see this is the spatial information, which have the top 2 principal components to be plotted, and every cell is plotted here as a dot. And for the heat map, you can also choose like top 10 or even top 20 genes to be seen here. And also, if you choose a balance, it always have a case control to be upregulated or downregulated scenarios here. In the 5th step, clustering, since our previous speaker already introduced a lot of these methods, in the Seurat, they are using the k nearest neighbor KNN graph with edges drawn between cells with the similar features expression patterns, and attempt to partition this graph into highly interconnected quasi-cliques or communities here. So for this, you need to find neighbors. For example, in the default, they have the 10 dimension. So from the PCA to find the neighbors. And after that, you can also do this clustering. They also provide this resolution. The resolution parameter size the granularity of the downstream clustering, which increase value leading to a greater number of the clusters. By default, they recommend to use value between 0.4 to 1.2, typically for a single cell data size of around 3,000 cells here. After you successfully do this fat clustering, it also showed how many number of the what number of the nodes, what number of edges here, and after you find the cluster, you can also run like run UMAP to do the dimension deduction and to see the further 2D cell cluster. So for here, and use the top 10 principal component to run the UMAP, and afterwards it detects this 9 cluster with the colors here. In the 6th step, differential express gene or marker identification, the goal here is to find out what's the difference between clusters. There are multiple ways, like a pairwise comparisons. You can define what the class A, what's the class B, and use the 1-to-1 comparison. Also, you can use one versus the multiples. For example, like this command, find markers, and give the identity of the to be compared is the cluster 5, and the left cluster is cluster 0 and cluster 3. Here you also have this minimal percent of the gene expression in each cluster. That means at least this gene have 25% to be expressed in both of these 2 defined clusters. You can also use a marker identification using 1 versus left, which you don't need to give the identification called ident.2. So therefore, it will automatically just give 1 versus the other. And after you run that, you can also have the view given this command hide, like the top 5 with the markers here. So you will also see the attributes like p-value, adjust p-value, average log 2 for change, and with the position of these genes in the 2D dimension. Afterwards, you can also visualize those markers genes here. So they provide like Weiling plot. You can see there are 9 clusters here, and once you select genes to be viewed here, it will just generate this Weiling plot with the distribution, expression distribution. You can also have a spatial information here, like if you just indicate how many genes that you want to visualize in that 2D plot, you can just give this as a feature, as input of the feature, and do this feature plot, and it will generate this graph for you. And if you know that the cell type corresponding to the markers, you can also give the identity of each cluster. For example, in the example, they have this marker IL7R and CCR7 that is usually highly expressed in the naive CD4 positive T cell. Therefore, they can assign the corresponding location of this expression called the CD naive CD4 T cells in this red color. The downstream analysis, for example, can perform the enrichment analysis and gene network. So in this example, we have used a pager database. Is a pathway annotated gene list, gene signature electronic repository for the human network biology. Here we have the interactive UBright2 web app I will show later in the real demo. So in the session 4, you can adjust the pager parameter to be to do the enrichment analysis here, and it will generate a table and also generate a network here. So firstly, I want to just introduce about the ATTIS from the Seurat. So here's the link. So in this ATTIS, they provide multiple panels, but if you're interested and also you have the data of the PBMC, you can do this analysis. In this specific panel is unique to the PBMC, so that will be regarded as the reference cluster, and you will upload your data to this website, and it will automatically generate those corresponding cluster with the cell type. So in the pre-processing, as I indicated before, that if you upload your data, I'm using the demo data, but if you upload your data, you can do the same filtering, like using the minimum, maximum, the range of the N counts of RNA, the range of the N features of RNA, and the range of this percent of the mitochondria. And also, you will just have the reference metadata to transform. So in this PBMC, they use a cell type I2. And after you upload your cells and your data, they will do this basically statistics and to check whether the quality is being passed. And afterwards, you can do the further cell plots. So this is a reference cell types in the 2D space. And this is what you query here. So I use their demo data. So they can also generate this percentage of the your original data and the target reference data cell type, and overall the cells percent in each cell type. Afterwards, you can also select the features and also do this feature plot in the 2D space, and also the volume plot to see the expression distribution here. So for example, if I'm interested in in the B intermediate cell type, they will just report the RNA biomarkers here, or the imputed protein biomarkers here. So for the top one, like the BANK1, it has a high AUC value at 0.932. So if I just choose this as the features, and you can see this is plot correspondingly in this region, and it has a high expression in the B intermediate cell type. And also you can see, because there's a mixture of some like B memory cells and B naive cells, therefore these three clusters also have this two other cluster also have a high expression of this BANK1 expression. Afterwards, it also allowed you to download the result. For example, for the ATTIS, you can download what the steps or what the procedure they have proceed in this demo. And they also provide those UMAP, imputed protein and predicted cell type and scores matrix here for you to download. Next, I will introduce about the UBright2 web app, which we have developed. We follow the general guidance and divide this downstream analysis into 5 sessions. So the first session is filter cells based on the quality control matrices. And you will see that we have this plot, N features RNA, N count RNA, and percent of the mitochondria. And we also have this sidebar, which allows you to set up the cutoff here. And afterwards, to visualize these highly variable genes, we also have this option to allow you to see the top N gene labels to be displayed in this standard variance plot. In the session 2 is a dimension deduction. We do the cluster of the cells based on the principal component analysis and UMAP. To visualize the, like in the 2-dimensional space, we have used the UMAP, and you will see here is the result. And afterwards, we also provide you marker for a specific cluster. So according to the this result, you can select the cluster you want to visualize, and also show the table, the top N genes represented in that clusters. Also, this table is downloadable using this link, and you can also visualize. You can also see those markers corresponding to each different cluster here. So if you want to show like top N, like top 10 markers in independent cluster, we have provide this table, and it's also downloadable. In the session 3, visualize this marker. Since you choose that's top 10, so we will just visualize in this heat map the top 10 across the 9 clusters. As well, we have this violin plot that you can select the genes that you're interested and to do additional visualization of this violin plot. If I change this parameter, it will rerun this graph and generate this plot accordingly. And the same, we also have this spatial information of this expression, the gene expression here. Afterwards is the input genes. Like if you select the cluster and choose the top genes, it will generate this table for you, and it also will run that pager analysis and provide this overrepresented analysis result. And it's also downloadable. For example, if I download that, I can see a file like this, because you may observe that some of the name have been truncated, but once you download, it provides a full gene site name here. Wait a second. Yeah, so Excel is loading here. Yeah, so this is the result. You can have a detailed view of all those information here. It has a source, gene site size, description, also the link, the type, the overlap, the code score, which we define in our publication in pager, similarity score, p-value, and force discovery rate. In the session 5, we have this generate the network of the selected pack. So according to the previous overrepresented pathways, you can select one of them and generate the network. It will also map what the cluster, like for here, it's from the cluster 0. So this genes is upregulated. Therefore, you will see this network here. And all those genes are the member of this pathway called the 1,4-dichlorobenzene degradation. And we also allowed you to download in the CSV files. And of course, you can use a third-party software like Cytoscape to do this visualization. And since our generation, the graph is static, but once you just export those files and import to this Cytoscape, you can have a full control of all these genes. And this is our works, how to cite our works. For the pager analysis, we have this paper, and for the protein-protein interaction information in network construction, we have this paper. And we also have the website. For example, this is our most updated pager, and it's also under construction for the future visualization panel. I will thanks to the faculties and scholars here in the Informatics Institute for their support. Dr. Minggao, July Wang, Dr. Dick Chen, Dr. Alexander Rosenberg, Dr. Chen Chong, and Dr. James Mino. Thanks. That's my presentation. Any question?
Thank you, Zongliang. Is there any questions? Thank you. Thank you for your attention.
Okay. Okay. I was going to ask this. You were now it's gone.
You're mute. You should.
Yeah, I wanted to ask you about the platform. Yeah, hello?
Hello. Yeah, we can hear you.
Right. I was trying to ask you about the platform you just presented. Is that in the Informatics Institute? It's in Jake's lab?
It's a platform. You mean the Zongliang introduced?
Yeah, the software he just talked about, yes. I
Yes.
know that Jake he developed the pager and PAGS, but now he has actually upgraded it to include the
Well, I think the voice has been cut off. Oh, sorry. Yeah.
Yeah, I was asking if the platform you just described for single cell analysis is in the AI lab.
Oh, yes. So currently this app for the single cell analysis, we are running it in the Streamlit and also implement our code here.
Okay.
Yeah. Also, we have the official website for the pager analysis, not limited to single cell, but also like bulk analysis or tissue specific gene expression.
I see.
Right. Yeah.
Okay, I'll link up with you later.
Okay. Yeah.
All right, thanks.
Thank you.
Other questions? Okay, thank you. Thank you, interested in the single cell workshop.